This lecture, we're going to focus on the plasma membrane. We will also talk about diffusion and osmosis. Last lecture, we talked about the eukaryotic cell and the organelles within the eukaryotic cell, but we didn't go in much detail about the plasma membrane. We have talked about how all cells are surrounded by a plasma membrane. It defines its borders, it outlines the cell, and it's also going to interact with its environment. We've talked about how the Golgi apparatus will package proteins and other things to leave the cell, and they're going to leave the cell through the plasma membrane. Some molecules and proteins and structures can move right through the plasma membrane, and then others are going to need specialized channels to help them get through the plasma membrane. Plasma membranes are made up of phospholipids. And a phospholipid, it consists of a phosphate group and a glycerol with two lipid tails. And these are going to be oriented in a specific way. So you can see all of these little balls at the top here. Those are going to be the phosphate heads. And then you can see the lipid tails hanging down. There's actually two layers here. This is called a phospholipid bilayer. And you can see the heads on the outside here, and then again on the bottom layer, the tail is pointing upward. They are oriented in that fashion because those heads are hydrophilic. So that means that they are water-loving, hydro-water-philic loving. And so that phosphate glycerol head is going to be oriented towards the liquid, the cytoplasm, the extracellular fluid, because it's hydrophilic. Fats, as you know, do not like water. If you put olive oil on water, it's going to float. So those hydrophobic tails are water-fearing, hydrophobic. And so those are going to be oriented towards the inner part of that phospholipid bilayer because they don't want to interact with the cytoplasm or the extracellular fluid. And then once you put them all together and you see this phospholipid bilayer, you can see that they arrange themselves so that those heads are facing towards the liquid, either outside the cell or inside the cell. And then those hydrophobic tails are going to be faced inward of that bilayer. And you can see the orientation here. This would be the inner cell, and out here would be the outer cell. And so any way you look at it, that phospholipid bilayer that makes up the cellular membrane is organizing itself based on these hydrophobic heads, hydrophobic tails, and hydrophilic heads. Because of this orientation of that phospholipid bilayer and how they are self-arranging, the membrane has some fluidity to it. It has some flexibility to it. And this is important because cells need to bend and move within the body. And this is a great depiction of the fluid membrane. And you can see it flexing and bending and moving. And these are those phospholipids self-arranging themselves based on the water, so based on being either hydrophobic or hydrophilic. There are a lot of different proteins and cholesterols and glycoproteins embedded in the membrane. Some of these help keep the membrane slightly rigid, slightly flexible, and some also help molecules and proteins pass through that membrane. Because there are so many different structures within the membrane, we call it the fluid mosaic model. It's almost like a mosaic of lots of different structures. The fluidity and all of the embedded structures within the membrane is what makes up the fluid mosaic molecule. And this is a semi-permeable membrane again. Some substances can pass through this membrane and some cannot. Larger substances will need channels like protein channels to fit through, and other smaller molecules like oxygen and lipids will be able to fit through on their own. And you can think of a semi-permeable membrane like this dog here behind this fence. And this fencing is a great example of a semi-permeable membrane because small things can fit through this fence. Little bees, oxygen and water, lizards, any other, maybe butterflies, but this dog here cannot fit through this fence. And so that is very similar to a semi-permeable membrane where smaller molecules can fit through, but larger ones cannot. Now there is a gate in this fence, and you could open the gate and allow the dog to leave. That's the same thing as the protein channels. The protein channels open up and allow those proteins to go either into the cell or out of the cell. Therefore, the cell can regulate what is coming in and out of the cell through those protein channels. Same thing as this gate, you can regulate where your dog is. You can keep your dog confined to the deck, or you can open up the gate and allow it to roam. When molecules move, they are going to move from an area of high concentration to low concentration, and this is diffusion. Diffusion is simply when molecules move from an area of high concentration to low concentration to reach equilibrium. And occasionally molecules can move freely. These oxygen and CO2 molecules can easily move across this membrane here, this phospholipid bilayer. And they will continue to move across this membrane until they are in equal concentrations on either side of the membrane. If there were more oxygen molecules on one side of the membrane, then it would diffuse to the other side of the membrane. On this little video clip, we can watch the diffusion of these molecules. At the very beginning, you can see that they are more concentrated at the top, and then through time, they will diffuse from this high concentration to lower concentration until they reach equilibrium. And this is called a concentration gradient. When you have areas that are more concentrated than others, that creates a concentration gradient until those molecules are evenly distributed. This is showing you that not all substances, not all molecules can fit through that semi-permeable membrane. So not all substances can diffuse across that membrane, but very small molecules can. When molecules can pass through the membrane on their own without needing any protein channels or anything like that, they can just move through that substance through the semi-permeable membrane, we call this passive transport. And that's just a fancy way of saying diffusion. Passive transport means that they don't need any energy or any help. These molecules are capable of moving through the semi-permeable membrane on their own from an area of high concentration to low concentration until they reach equilibrium. There are three main factors that will affect diffusion, or the rate of diffusion. How strong the concentration gradient is will affect the rate of diffusion. So if you have a very high or very strong concentration gradient, or where you have a greater difference in concentration, so you have a lot more molecules on one side of this membrane than other, then you will have a more rapid rate of diffusion. So the higher the concentration gradient, the more rapid the rate of diffusion. The size of the molecules also has an effect on the rate of diffusion. Heavy molecules are going to diffuse slower than lighter molecules. So the mass of the molecules is important when considering the rate of diffusion. And finally, temperature is going to affect the rate of diffusion. Temperature is really just of the movement of molecules. And at higher temperatures, molecules are moving faster. At lower temperatures, molecules are moving slower. So at higher temperatures, molecules will diffuse faster than at slower temperatures. So these are the three main conditions that affect the rate of diffusion. We mentioned and we saw before that not all molecules will fit through the membrane. And because of this, we have something called osmosis. And osmosis is the same thing as diffusion, but it involves a semi-permeable membrane. And so osmosis is just diffusion through a semi-permeable membrane. Some molecules can easily pass through the membrane. Lipid molecules, oxygen molecules can easily pass through the membrane. But larger molecules like glucose, salts, things like that, will have a harder time fitting through the membrane. And therefore, you will end up with a concentration gradient where you may have more solutes on one side of the membrane compared to the other. And this will affect the water concentration or the cytoplasmic concentration on either side, depending on where those solutes are. When we talk about solutes, we're just talking about the substances that are dissolved in an aquatic medium. So here is a cell, and this cell, this is the plasma membrane, the semi-permeable membrane around this cell, and that's why it's dashed. And the solutes, you can see, are sodium and chloride. And there are some solutes both outside the cell and inside the cell. Those solutes are dissolved in a solution. The solution is the aquatic medium. In this case, it's water. And you can see that this cell here has 5% solutes inside the cell, which means that there is 95% solution inside the cell. Outside the cell, the extracellular fluid has 10% solutes, which leaves 90% solution. Because the solute solution is higher outside the cell than inside the cell, water will end up leaving the cell to try to reach equilibrium, because remember that these solutes cannot fit through this semi-permeable membrane. Only these small water molecules can fit through the semi-permeable membrane. And so because there are more solutes outside the cell than inside the cell, and the water concentration is greater inside the cell than outside the cell, the water concentration is going to try to reach equilibrium. It is going to try to dilute the solutes outside the cell. And we call this hypertonic. So when the solute concentration is higher outside the cell than inside the cell, the water will leave the cell, and the cell is living in a hypertonic solution. Here we have a similar cell, but our solute concentrations are different. In this cell, we have 10% solutes inside the cell and only 5% solutes outside the cell, which leaves us 90% water inside the cell and 95% water outside the cell. In this situation, this cell is considered to be hypotonic. The solute concentration is lower outside the cell than inside the cell. And because of this, water will go inside the cell. That will cause the cell to swell. The hypertonic, recall, cell will lose water and will shrink. The hypotonic will grow and take on water. And again, this water is trying to reach equilibrium. Remember, only the water can fit through the membrane, not the solutes. And so the water is trying to reach equilibrium. The water is going to go where it's high concentration to low concentration until it reaches that equilibrium. And lastly, we have a cell where the solute concentrations are equal inside and outside the cell. And we call this isotonic. You can still get net movement of water across the membrane, but you're not going to get any net change because the concentrations are the same. So water molecules can still move back and forth, but you're not going to get a change in any of the concentration because there's no concentration gradient here. They're equal. So that is called isotonic. Again, hypertonic cells are going to lose water and shrivel up. This is a plant cell here. Remember, we talked about that central vacuole in plant cells, and that is what is giving up its water in the hypertonic cell. The cell wall is going to help maintain rigidity within that cell, but that inner part is going to lose water and shrink. In an isotonic solution, again, you get movement of water into and out of the cell, but you're not going to get any net change. And then in a hypotonic condition, that central vacuole is going to swell up and take on more water. Some cells could burst. A plant cell won't burst because it has a cell wall, but some cells that don't have cell walls could potentially burst in hypotonic situations. And this is just another way showing you that cells are moving from an area, or molecules are moving from an area of high concentration to low concentration. Cells are not moving. And it's the water that's going to be moving through that semi-permeable or selectively permeable membrane. And they are going to be affected by the solute concentration across that membrane. The higher the concentration gradient you have, the more rapid change you will get, and the lower the concentration gradient, the less change you'll get. Some solute molecules will need help getting across that membrane, and we call this facilitated diffusion. It's similar to diffusion, but it has these open channels that the solutes can move through freely without being affected by that semi-permeable membrane. This is also known as passive transport because it does not require energy to move those solutes through that transport protein. That transport protein can open and close as the cell wants to accept solute molecules or not accept them, or allow them out of the cell as well. But these solute molecules are going to continue to move along the concentration gradient. They will still be affected by that concentration gradient, and they will still be moving from an area of high concentration to low concentration. Some molecules need help getting across a membrane, but they need to go through a transport protein that requires energy to open and close it. This is an example of a neuron in your nervous system. And in order to get the potassium and the sodium across the membrane, the cells use these proteins that require energy. And so you have your transport protein, and the solutes can bind to that transport protein, and then energy allows that protein to open and expel its contents on the other side of the membrane. And then it will return to its other shape. And we call this active transport because it requires energy input in the form of ATP. ATP is the energy currency that cells use. It is made through the process of cellular respiration, and we will learn all about ATP in just a bit.